Hello, my name is Lauren Velie, and I'm a member of the Vierling Lab at the University of Massachusetts Amherst. Today I'll be presenting on my project Unraveling the Role of AKR4C Proteins in Nitric Oxide Homeostasis and Arabidopsis thaliana. I'll begin with a brief introduction to the goals and methods of this project. So the long-term goal is to understand the role of these AKR4C oxidoreductases in nitric oxide homeostasis in the model plant organism Arabidopsis thaliana. We have two general approaches, first of which is to generate AKR4C knockout lines by CRISPR-Cas9 mutagenesis to eventually examine the phenotypes of losing multiple AKR4C proteins. Secondly, we are expressing and purifying AKR proteins for use in activity assays to better characterize their activity and substrate specificity. So first, some basic background. Nitric oxide is a key signaling molecule in plant development, as well as in humans and organisms at large. And Arabidopsis specifically, it is involved in shoot development, root growth, fertility, somatic closure, immune response, and seed germination. These are all processes vital for both normal plant development and stress response, including heat stress and also drought stress. So Gstr is the regulator of nitric oxide homeostasis inside of plant cells. So nitric oxide is a short-lived radical molecule and is integrated into S-nitrosoglutathione, or GSNO, as the bioactive form of nitric oxide in plants and many other organisms. So GSNO reductase, GSNOR, metabolizes GSNO and thus regulates endogenous nitric oxide levels, moderating the signal and cellular responses to the presence of nitric oxide. It utilizes NADPH as a coenzyme in this metabolism. So the second component of this project is examining aldoketoreductases. So the aldoketoreductase protein superfamily is characterized by, as we can see to the right of this slide, I have AKR4C8 pictured here, by an alpha beta 8 barrel fold, and substrate specificity is offered by loop A, loop B, and loop C, which are highly variable regions. They are monomeric NADPH dependent oxidoreductases in comparison to GSNOR's NADH dependent activity, and they metabolize a broad range of reactive carbonyls produced during periods of stress. Most importantly, specific mammalian AKRs, specifically AKR1A1 in humans, have been shown to metabolize GSNO, which links these aldoketoreductases to nitric oxide homeostasis in plants. So, other enzymes involved in nitric oxide homeostasis. So I have a volcano plot to the left of the slide. To the right of that spike in the middle are upregulated proteins in our GSNOR deficient, that is lacking GSNOR, unable to modulate GSNO levels inside of the cell via the use of GSNOR, compared to wild type expression. So we can see that these two, AKR4C8 and C9, are significantly upregulated. We see they have a positive log fold change in these GSNOR deficient mutants. So combined with this, the fact that GSNOR deficient plants show an increased NADPH dependent GSNO metabolism, we can hypothesize that this NADPH dependent pathway may be integrated with GSNOR, potentially a compensatory mechanism to make up for the loss of GSNOR in these plants in utilizing these AKR proteins to metabolize GSNO. And to the right, I have a graph exhibiting that NADPH dependent GSNO consumption is significantly increased in these HOT5-2 GSNOR knockout mutants. In addition, 4 AKR4C proteins, C8, 9, 10, and 11, have been shown to reduce GSNO. So, why would we want to knock out these genes? So the function of these 4 Arabidopsis AKR4C proteins is likely redundant due to their high sequence similarity. It ranges from 64 to 90% identity over similarity. And as a result, knocking out one of them will likely not generate a strong phenotypic response. So knocking out multiple, or hopefully all of them, will generate a phenotype representative of the loss of this family. In addition, CRISPR is the ideal component or pathway to get to this multiple knockout mutation, since Theoretically, using 2 guide RNAs per gene can generate a large deletion by way of introducing double strain breaks on either side of a region that will be deleted. This is also site-specific, so we can target multiple genes on the same chromosome. As we see in this figure, AKR4C8, 9, and 10 are all on chromosome 2, so the only way we can get homozygous knockout mutants is to generate mutations in all 3 at the same time. So we avoid needing to generate single mutants and crossing them and hoping to find some homozygous recombinant, which is highly, highly unlikely due to their very, very close physical proximity. So far, knockout lines for 2 of the AKR4Cs have been generated, AKR4C8 and C10. We have lines with deletions for each of those, and they are Cas-free, which means that the CRISPR-Cas9 construct is no longer within the genome of the plant, which eliminates the possibility of any off-target background mutations. So when we examine these plants for phenotypes, we are seeing hopefully just the effects of losing these specific genes. They have been backcrossed and are being prepped to have additional CRISPR constructs integrated to induce deletions and knockouts in each of the other 2 AKR4C genes, C9 and C11. So the second component of this project is working with the enzymatic function of these proteins. And to do that, we are expressing and purifying them. So they are expressed in a his-sumo-tagged construct into low background strain E. coli and isolated and cleaved to yield the final protein product, which can be seen in this SGS page. So to the left, we have the AKR4C8 his-sumo-tagged version, which is at around 50, a bit above like 55 kilodaltons. And to the left, our top red box, we have AKR4C8 that has been cleaved from that his-sumo-tag, and then flow through 2, which I have highlighted in blue, shows the cleaved and purified protein product, which indicates a successful protein purification. So this is a protein that we will use in future for activity assays to examine the substrate specificity and general activity of this enzyme in particular. We are expressing the human AKR, a catalytically dead variant of the human AKR, and catalytically dead variants of all of the AKR4C proteins that we are examining in order to have them as negative controls against functional wild type versions while setting up these activity assays. So the future directions involve examining the phenotypes of AKR4C8 and C10 knockout lines. Again, we do not expect any extreme phenotype since they are pretty redundant in sequence and likely function. We will confirm that these mutant alleles are null mutants through Western blots, just confirming that these proteins are not being successfully expressed within these plants. Additionally, we will utilize CRISPR-Cas systems to generate mutants with multiple knockouts in these AKR4C genes to eventually yield a complete knockout of this group of proteins, eliminating C8, C9, C10, and C11 at once to finally compare with our Hot5-2 Gstr deficient mutants and generate a phenotype or an understanding of their phenotype through that. And finally, we utilize the purified proteins and activity assays to test enzymatic activity and substrate specificity. Thank you so much for watching.